Swayam Prabha. Digital India. Educated India.
Welcome back to the lecture series in animal physiology. So we are into the week 7th and today we'll be starting the 5th lecture of this week. So in this week, if you look carefully, we have talked about Alzheimer's, degenerative disease of the brain. We talked about Parkinson, another degenerative disease of the brain which leads to movement disorder, contrary to Alzheimer's, where it leads to problems of self, losing memory about your own self and the surrounding. Then we talked about a degenerative disease affecting the ventral horn motor neuron, ALS, amyotrophic lateral sclerosis, and we kind of talked about the similarities and dissimilarities. Followed by that, we moved on to the spinal cord injury and we talked about the development of syrinx and how that prevents the further regenerative ability is being compromised and preventing further the axon to guide through that injured site. Today we will talk about the other cell type, the glial cells. So let's get back on the board. Today it is week 7th, lecture 5, W7L5. So we'll be talking about glial cells. So glial cells could be classified into two categories broadly, and we'll talk about those which concerns us the most. One is the Schwann cells, and the other one is the oligodendrocytes, whereas the third one is astrocytes. So these two, the Schwann cells, one which I am putting red boxes, these ones are involved in myelination, whereas astrocytes on the contrary is involved in homeostasis of the nervous system. What does that mean? That means, say for example, let's take a hypothetical situation. Inside the brain there are synapsing bodies and there are a lot of neurotransmitters which are getting released. So the oligodendrocytes, now I'll put them in the green, they are sitting like this. They are numerous, far more as compared to the neuronal cells. Their number is far more higher. They are the ones who ensures that hyperexcitability due to neurotransmitter being accumulated in the synapse is being prevented. They are the one who helps in recycling the neurotransmitter. They are the ones which, recycling NTs, neurotransmitters, they are the ones which provide nutrients and they maintain the homeostasis in terms of ionic level around the neurons. And many a times, because of their pathologies, these cells kind of divide uncontrolled manner and they could lead to form of tumors. These cells are electrically active. They shoot certain electrical currents, but they do not shoot an action potential, and their membrane voltage could never overshoot the zero. So if this is the zero and this is in millivolt I'm talking about, and these cells are sitting in minus 80, they will shoot something like this. You will see them, but just in and around, minus 60, minus 30, minus 40, but they will never overshoot the zero. So this is the zero line. So strictly speaking, if I say that they are not electrically active, that would be wrong. They are indeed electrically active, but do not shoot action potential. And the difference between them and the neurons are these cells do divide, and their uncontrolled division may lead to tumor in the brain. Second, they help in recycling of the neurotransmitters, provide nutrients, and maintain the homeostasis or the ionic balances. Now comes about the other two, the Schwann cells and oligodendrocytes, and the concept of myelination. So whenever we draw a classic neurons, the picture is something like this. Something like this, okay, with the cell body, the nucleus sitting here, and on this you observe something like this. Both sides something like this. There is small gaps out there. We'll talk about those gaps. Something like this, further this, further this, and this continues like this, okay. Okay, now this in a textbook you will see it is written as myelination, and this spot what you see, it's called nodes of Ranvier. Nodes of Ranvier. This one, this one, this one likewise. And nodes of Ranvier have very high population of voltage gated sodium channels, and the action potential kind of has a saltatory mode. It looks like it is jumping like this. Now action potential could have traveled all along like this if there would not have been, and it actually travels like that. But the thing is that, say for example, this green cover, or sorry, the blue cover would not have been there. Say for example, I draw something like this, this one, and the action potential, so this is one axon, say for example, of one neuron, and there is another neuron side by side moving, because these are nerve nets and nerve tubes which are moving, and if these are close by and there are no covering, so the excitability which is happening here will affect the excitability here. Excitability happening here will affect the excitability here, and not only that, in that process there will be information which will be lost like this. In other words, you can call this as a situation of short circuit. And that way, the information which from here, say for example, to reach here needs say x unit of time, will now take or may not even reach from here to here because of loss of signal. It has its advantage and it has its disadvantage. In terms of the disadvantage is your transmission slow, loss of signal, loss of signal, and compromising the efficiency of info transfer. So there are these disadvantages. Sometimes advantages are also there. A signal which harm or pains the system needs to be dampened. Okay? So this is a classic situation where loss of signal or dampening of the signal could come very handy, and as a matter of fact, in the case of pain, which is very excruciating pain, you want certain signal to get lost. You do not want the brain to kind of get bogged down by that heavy excruciating signal of pain. And there is a fundamental difference between those neurons which carry the pain signal as compared to the neurons which are conveying other kind of signals. The one which are carrying the pain signals are unmyelinated neurons. They do not have any such cover, just like this. Contrary to it, you have these myelinated neurons out here which cannot afford any kind of these disadvantages. They cannot afford it, right? So myelination is a process by which there are categories of cell which are termed as Schwann cells and oligodendrocyte, which forms an insulation on top of these axons. Now how they do form? So myelin is a protein which is secreted by these cells. So the way it works is very interesting. When the axon is forming in mother's womb, so for example this is the axon of a neuron. Now the Schwann cells, we'll first talk about the Schwann cells, then I'll tell you about the myelination with the oligodendrocytes. Okay, these are the dendritic trees. Here you have the nucleus setting. So the way it works is the Schwann cells started to migrate at the site of myelination. They all goes there and they started to align themselves like this all along. Okay, both sides they are coming, say for example, okay, like this. Then there is a second level of process which occurs. These cells divide. They started to divide like this. And interestingly, some of these cells post division kind of goes underneath the axon. So say for example, if this is the axon these cells will go almost underneath. So some of the cells will be underneath it, and some of the cells will be top, and they started to climb on top of each other. So say for example these cells, they started to climb like this, and through this climbing motion on both sides along this rod, say for example if this is the axon, so these are the glial cells, so they eventually form a kind of a coating like this, something like this all around it, and then these cells slowly lose their individual identity and they merge. So these individual cells eventually merge and they secrete a protein which is myelin basic protein. These protein along with this cellular structure forms what I drew in the previous slide, what we call as the myelin sheath. It's a very controlled process. What we do not know, absolutely no clue, how they decide that where they have to have these nodes of Ranvier. Who decides, how they decide, it's still a mysterious thing. But they do divide in a most beautiful controlled way, by virtue of which they create this kind of very interesting pockets where the insulation is not present, and that is the place where the external excitability kind of access the axon. Okay, so this is the role of the Schwann cells, but several unfortunate individuals, in their case, because of certain autoimmune situation, these Schwann cells started to die, and therefore if they die, then of course in nature there are situations where there are neuronal pathways which are unmyelinated. So we have two categories of pathways which are unmyelinated and myelinated. Okay, so this has been already decided, but imagine because of a disease situation these myelinated becomes unmyelinated, because these Schwann cells started to die out, and this is a situation what happens in multiple sclerosis, where the insulation of the myelinated neuron is being compromised, which leads to severe problem. It's partly genetic, and there are many things which you do not know about it, but that's one issue which affect the patient heavily. There are different kind of antibodies which are there against that autoimmune disorder. There are people use. So, but still, it's a very, very compromised lifestyle. And the Schwann cells are present outside the central nervous system. So the myelination of the neurons. So for example, now let me again redraw the spinal cord and the brain. So whenever we draw it, we drew it like this, right? So, okay. So now the neurons are sitting here, right? And they are sending out the processes. So all the myelinations which are happening, they are happening outside. And this is the myelination of the Schwann cell myelination, right? But as soon as this neuron enters inside the spinal cord and goes to the brain and wherever, the myelination pattern changes. Same neuron is myelinated out here by another set of myelinating cells, but they myelinate in a different way. Those are called the other cell type called oligodendrocyte. oligodendrocytes. So a neuron whose part of the body or part of the axon which is inside the spinal cord and part which is outside the spinal cord will be myelinated differently. The one which is outside, so that the location out here, will be myelinated by Schwann cells, whereas the one which are inside the spinal cord will be myelinated by the oligodendrocyte. But oligodendrocyte myelination is very different. Oligodendrocyte does not follow the paradigm followed by the Schwann cells. The way oligodendrocyte myelinate is very interesting. Say for example, this is inside the brain somewhere or spinal cord. These are the processes I am talking about. Cell body sitting there, and so the way these myelination happens is something very interesting. So here you have numerous oligodendrocytes, and these are oligos are like this. There are thousands of oligos in and around. These oligos send out these processes like this. Philopodia or processes. So they form something like this. So this is where oligodendrocyte forms. So one oligodendrocyte can myelinate 10 or 12 or even more neurons at one point of time. Why nature has done it? Probably to save space, unlike because brain has, so you see the same oligodendrocyte, it is myelinating here, this is myelinating here. It can even, if you go down the plane, it can myelinate something else. So these are the zones of myelination. Okay, so oligomyelination is entirely a different kind of pattern, unlike the Schwann cells myelination. So what I was telling you about the space. So here you always have a space limitations. You really cannot increase the size of the brain, and you cannot really spread it. So out here, possibly nature has divided or designed this in such a way that a single oligo can, so it's something like this. So for example, if this is one oligo sitting there, this oligo will be forming like, like, it's almost like you have seen that Spider-Man, this thing, something like that. So even if you look at it, at one point, this oligo just in one plane could, myelinate 1, 2, 3, 4, 5. So there's another oligo, say, sitting out here will be myelinating that axon, or another one coming like this axon. So it's a very interesting network of oligos which are doing this job in your brain. So this brings us to a full circle of talking about the different types of Schwann cells, and I'll request you please go through standard textbooks which are there, but the crux of the matter is what I have taught you, that you have Schwann cells, you have oligos, you have microglia, but the microglia's origin, as I have mentioned, are mostly from the immune cells origin. You have the neurons of different types. They suffer from different kind of neurodegenerative disorders, whereas there are brain tumors caused due to the glial cells. There are degenerative disorders of the glial cells because of certain mutations or autoimmune aspects which leads to multiple sclerosis. So that kind of brings us a full circle about the different cell types constituting our system. Next, what we will do is we'll talk about the special senses, which will be including eyes, ears, tongue, skin, and we'll talk a little bit about stretch reflex at the end and neuromuscular junction. So with this, I'll conclude this week, and thanks a lot, and please do read.